Uh, in this handout, we're going to be touching upon topics that correlates with these learning objectives. We're going to start off with the factors that affect GI absorption. GI means gastrointestinal absorption, things that get absorbed through the gastrointestinal tract once it's given orally. The thing to remember is systemic absorption is favored after oral absorption because the body acts as a sink. That means the body has very minimal, if at all, very minimal amount of the drug that is being introduced into the body through the oral route. The GI tract has the most amount of drug because that's where the oral dose resides during the absorption process. And so there's a huge concentration gradient that establishes between the GI tract and the systemic circulation. So naturally, systems tend to equilibrium, and so material will get absorbed, provided that there are adequate conditions exists in the GI tract. Provided adequate conditions exist in the GI tract, we have all things naturally present to favor absorption. Now, there are two very important determinants of gastrointestinal absorption, primarily gastric emptying and intestinal absorption and permeability. The first potential site for absorption of orally administered drugs is the stomach. And in accordance with the prediction of the pH partition hypothesis, which basically tells us that acidic drugs tend to be absorbed to a greater extent from the stomach as opposed to small intestine, because in the small intestine it is less acidic, and hence it favors absorption of less acidic drugs because obviously for transmembrane permeation or transmembrane absorption to occur, molecules will have to be more in the unionized state, and so depending on the pKa of the molecule and the pH of the environment, the degree of ionization of the molecule can vary, and hence the region that favors greater unionized molecules will lead to greater absorption of that particular unionized molecule. I mean, that's what is pH, what is meant by pH partition hypothesis. And so weak acids are absorbed primarily very rapidly from the stomach, as opposed to the small intestine. However, gastric emptying is an important determinant. Food, especially fat, can slow gastric emptying, which explains why drugs are frequently recommended to be taken on an empty stomach when a rapid absorption and a rapid onset of action is desired. Drugs that influence gastric emptying also can affect the speed of absorption of other drugs, and I have some examples and graphs and data to support this statement later on. Additionally, intestinal absorption permeability, because of the fact that the intestine and the length, throughout the length, the properties of the intestine varies in its various properties as well as luminal composition throughout the intestine. This is an important factor. The intestine may be broadly divided into the small and large intestines, separated by the ileocecal valve. Surface area per unit length, one parameter that can be determined, decreases from the duodenum to the rectum. And additionally, electrical resistance, a measure of the degree of tightness of the junctions between the epithelial cells, is much higher in the colon than in the small intestine. And hence, only water, if at all, gets absorbed from the contents that are brought into the colon region. Now, there are several causes. Even though the environment, nature, the physiology and anatomy favors absorption of material from the GI tract, there are several causes for loss of bioavailability. There are several factors that are listed there. We will go into detail of each one of those as we navigate through these slides. One cause for loss of bioavailability is competing intestinal reactions. We will talk more about that. Transporter effects, gut wall metabolism, and first pass loss. Additionally, the the model, as well as many of the factors that we discuss in this, except for the pH gradient stuff, applies to absorption of drugs through the intramuscular that are introduced into the body through the intramuscular and sub-Q routes as well. This applies to absorption from solid dosage forms, and we will also talk in detail about the biopharmaceutic classification system, which actually categorizes drugs based on their permeability and solubility into 4 general categories. Okay, as far as understanding oral absorption and trying to delineate and focus on factors that can affect absorption, the best model to start with is actually not a solid dosage form but rather a liquid dosage form, because passage of drugs through a membrane dividing the absorption site from the blood is a prerequisite for absorption, and this process occurs naturally and much faster from a solution. Even if it's a solid dosage form such as a capsule or a granule or a tablet that is being introduced into the GI tract, the drug will have to dissolve in the GI contents or the water that is being taken with the dosage form before it can actually get absorbed. So the drug will have to be present in the monomolecular dispersion form. That means it has to dissolve completely entirely in a liquid to be able to absorb through the membranes. Most oral drugs, however, we know are not administered as a solution. Majority of the drugs are solid dosage forms, but as I mentioned earlier, these solid dosage forms will have to disintegrate and the drug will have to dissolve in order for it to be absorbed. So first we'll start with absorption from a solution. I already mentioned this. Systemic absorption is favored because the body acts as a sink. The amount of drug that is present in the GI tract is much greater than what is eventually going to reach the systemic circulation. So there is this huge concentration gradient that is established, and that concentration gradient actually favors absorption of molecules across the GI tract. Additionally, there are some physiologic and physicochemical factors that actually can impact movement of drug through the membranes, molecular size of the drug, additionally, nature of the membrane involved, presence of transporters, perfusion, and of course pH, because pH is connected to the degree of ionization, as well as the fact that drugs that are absorbed through transmembrane diffusion, transmembrane absorption, the molecules will have to remain in the unionized state to favor absorption through that mechanism. Okay, and those are some of the factors, physiologic and physicochemical factors that can impact movement of drug molecules through the GI tract, through the membranes lining the GI tract. Now this diagram kind of shows the different regions of the GI tract. You're familiar with this. This diagram figure is there to show you the complexity of the regions involved and the categorization of these regions within the GI tract, so that you can actually place the points that are going to be mentioned throughout this lecture in the right perspective, in the right context. Okay, so familiarize yourself with the diagram, the general diagram that shows the gastrointestinal system, different regions, and how they connect and relate to another part of the system. Okay, stomach. We'll start with stomach. This is the first potent site from where absorption can occur. Now, I mentioned pH partition hypothesis earlier. So what pH partition hypothesis is all about is that molecules that are acidic are going to be less ionized in an acidic environment, and hence will be rapidly absorbed from an acidic environment. And so stomach that is highly acidic when compared to small intestine will favor absorption of acidic, slightly acidic or weakly acidic molecules. That's what the crux of that statement, pH partition hypothesis term, means. Absorption of acids is much faster, though, from the less acidic small intestine than from the stomach, and that's because of the fact that there are other factors. Although pH partition hypothesis hypothetically leads us to assume that acidic molecules will get absorbed from the stomach faster than small intestine, there are numerous other factors that will negate that effect, such as surface area, permeability. All of this will contribute to greater absorption no matter what the type, nature of the molecule, whether it is acidic or basic. In the GI tract, because of various physiologic and anatomic factors that come into play, molecules will get absorbed at a much faster rate from the small intestine than the stomach, even if the molecule is acidic. And that's what the three main factors that actually favors absorption of molecules from the small intestine is actually shown in that table. For example, if you were to compare the absorptive area available in the stomach, it's about 1 meter square. However, in small intestine is about 200 square meters. Blood flow, things that are getting absorbed should be able to be removed from the site of absorption, right? And that occurs through blood flow, blood perfusion, right? So in the stomach, blood flow is about 150 milliliters per minute. In the small intestine, it's 1 liters per minute. Okay. Now permeability, the membranes, the structure, as well as the anatomy of the structures lining the stomach makes the membranes low permeability. However, small intestine, very porous. It favors absorption, and hence, in general, permeability is high. The membranes have higher permeability in the small intestine. So these three factors contribute to the greater absorption that is typically observed amongst all kinds of molecules in the small intestine. All right, we'll start with gastric emptying. We know food slows gastric emptying, particularly fatty foods, and this is the reason why sometimes drugs may be, except for when the drug has an irritating effect in the stomach, or when it's taken alone, and an empty stomach can be harmful to the stomach membranes. That's the only time when you actually would take it with food. Otherwise, it's always recommended that you take a drug in an empty stomach, and particularly if you so desire a rapid onset of action. Now, the influence of gastric emptying on drug absorption is what is shown on the slide. Gastric emptying can, in instances, be the rate-limiting step for systemic drug absorption from the GI tract. For an example, remifentanil, which is an ultra-short-acting opioid narcotic analgesic, it actually slows gastric emptying. We know this is true, right? Of a lot of pain medications taken orally will slow down, Percocet, for example, OxyContin, for example. These actually slow gastric emptying or gastric movement, GI movement in general, and that's one of the side effects associated with daily intake of this type of medication, right? It slows down the GI tract movement and leads to development of constipation. And so that actually can have an impact on how drug gets absorbed in the GI tract when such analgesic, narcotic analgesics are taken orally. What is shown here is actually remifentanil, an ultra short-acting opioid narcotic analgesic. How it slows down gastric emptying is demonstrated in the slide. the drug was given alone, and that is what is shown in the black lines. And 10 minutes after the start of a constant rate infusion of remifentanil in the colored lines. And the posture, when the person was asked to lie on the left side as opposed to right side, was also determined in this study. So what you see actually here is when the drug is given alone, and of course, what was being studied was acetaminophen absorption from the GI tract when remifentanil was actually administered. So uh, when when the drug was given alone, that means when acetaminophen was given alone, that is what is shown on the black lines. So the right side, when the person was lying on the right side, the peak concentration was attained faster. And if you look at the AUC, you seem to see that greater bioavailability can be seen on the right side as opposed to left side. I mean, that has to do with the natural anatomy. I mean, how if you lie on the right side, contents in the stomach can be expelled into the GI, into the intestinal, into the duodenum faster, as opposed to when you're lying on the left side, it has to be ejected forcefully ejected opposite. So it's slightly, I mean, although it's not going to impact much, of course, you're seeing here a slight impact on the posture here. Now, when it comes to the red lines, that indicates when acetaminophen was actually given along with remifentanil. And as you can see, the plasma levels of acetaminophen has drastically reduced, and that's because remifentanil slows down the GI tract. And so acetaminophen absorption is clearly impacted. A clear demonstration of how narcotic analgesics impact gastric emptying, and how that can impact drug absorption from the GI tract. Now, intestinal absorption permeability, one parameter I mentioned earlier that can be followed through the GI tract is actually the surface area per unit length, which tends to decrease from duodenum to rectum. Additionally, electrical resistance is very high in the colon because of the tight junctions that exist between epithelial cells that actually line the colon. And this actually leads to less, if not completely, complete resistance to absorption of molecules from the colon, and hence, small intestine naturally favors absorption of molecules from that area. Now, proteolytic and metabolic enzymes and active and facilitator transport systems are also further expressed variably along the intestine. And so there are some regions of the intestine where you would see a preponderance of transporters when compared to other regions of the intestine, and hence drugs tend to be absorbed from that particular region much, much faster. The permeability surface area product, as I mentioned, progressively decreases from duodenum to colon, and this applies to drug molecules traversing the intestine epithelium by non-carrier mediated processes, particularly through the transcellular permeation process, transmembrane diffusion process, and also, or paracellular routes when drug solutions are placed in different parts of the intestine. Okay, now, as far as the case of permeable drugs, drugs that naturally permeate through the intestine, absorption is rapid and perhaps incomplete within the small intestine. But even if a small fraction is not absorbed or from the intestine and reaches the colon, most of the drug is completely absorbed, in the small intestine. The absorption across intestinal epithelia may also be perfusion rate limited. If the absorption is perfusion rate limited, that means, obviously, the faster the drug gets to the intestine, faster and greater will be its bioavailability, because we know that from the earlier table that compared the perfusion rate between stomach and the intestine, we saw that a small intestine had greater blood perfusion, and hence the faster the drug gets to the small intestine for those drugs that are perfusion rate limited, absorption is perfusion rate limited, then it better get fast to the get to the intestine. And hence, for such kind of drugs, the overall rate limiting step in systemic absorption is going to be most likely how fast the drug is going to be emptied, how fast the contents from the stomach is going to be emptied into the small intestine. Now, molecular size is really important when it comes to membrane permeability, particularly small polar molecules. Some examples, is cyclovir. The molecular size is 250 grams per mole. The H2 antagonist cimetidine, 252 grams per mole, and atenolol, 266 grams per mole. These primarily move across the intestinal or GI epithelium through paracellular route. So what is paracellular route? This is the absorption mechanism through which small molecules, such as the ones that are listed there, get absorbed by being in a solution in water in an aqueous medium, and then it is capable of going through the pores, small pores and gates that line these epithelial cells that are present in epithelial cells. So that route, the absorption through that route, however, or rather the permeability in general through that route, paracellular permeability in particular, will drop off sharply with molecular weights about 350 grams per mole. And a value at which compounds appear to approach the molecular dimension of the tight junctions between the intestinal cells. So these pores that line these tight junctions, as well as that are spread around all across the epithelial cells, they tend to match or become similar to the size of the molecule when the molecular weight is about 350 grams per mole. And so those molecules that are greater than 350 grams per mole will have great difficulty to be absorbed through that route. When the molecules are smaller than that size, paracellular permeability favors absorption. Just by virtually being in solution, water that goes through the pores will carry the molecule to the other side of the membrane. Here's a very interesting data that actually shows the fraction of a dose that is intestinally absorbed after oral absorption versus human jejunal permeability. So the percent of dose that is intestinally absorbed is plotted in the y-axis, and jejunal permeability in centimeters per second times 10,000 is actually plotted in the x-axis. So it's a way of looking at how molecular size can actually correlate to permeability. So what is shown here is drugs with permeabilities less than 1 times 10 to the power negative 4 centimeters per second are likely to be incompletely intestinally absorbed. So right around there, right? You see that it kind of the graph kind of plateaus off here, but it actually seems to have a definite trend up to about that point, right? As you can see by that blue line that I just drew over that. This actually compares the jejunal permeability of various compounds, jejunal permeability of various compounds that are listed in the graph when it was compared or studied in terms of its percent of dose that was absorbed when given orally. And studies like this can actually give us more information about what mechanisms are prevalent in terms of drug molecule permeability from the small intestine, and you know these types of studies will help us to understand which type of molecules will eventually have a problem when given orally. How can you address that problem, right? So this type of studies will help us to understand that, that aspect of intestinal absorption. So you know, what it shows is when the drugs have low permeability, those are the drugs that are to the left of the value of 1, um, marked in the x-axis. So clearly, low permeability drugs are likely to be poorly absorbed because, those drugs have very low permeability. Even enalaprilat, fexofenadine, methyl-dopa, all of those when compared to propranolol and desipramine and losartan, or even metoprolol, they have less permeability and hence a fraction of dose that is absorbed. Right? For example, enalaprilat permeability is very low, and so what is it? I mean, if you look at this value, less than 10%, right, it's going to be absorbed from an oral dose that is administered. So you clearly see the correlation between intestinal permeability and the dose that is intestinally absorbed. As far as more examples that show this type of pattern in terms of oral absorption and permeability, some examples are listed there. These particular drugs have very low intestinal permeability and hence low oral bioavailability. Alendronate, amikacin, carbencelin, cefamandole, ceftazidime, all of these molecules have low oral bioavailability because of their poor or low intestinal permeability. Now, intestinal absorption and permeability of less permeable drugs. Right? Often, these less permeable drugs are often more polar, and in such instances, absorption primarily still will occur from the small intestine rather than the large intestine, as is shown in the diagram. So, what is shown there is actually comparison of the gastrointestinal absorption of ranitidine depending on the site of application. So a study like this can be done in animals. You actually can also do this kind of study through in human beings. A study like this in vitro can be done by actually excising regions, stomach, jejunum, and colon, surgically obtaining these regions from animal models and actually studying that way. Or actually, you can actually do surgery and perform the studies in animals by placing the drug through tubes placed in specific parts of the GI tract. In human beings, we can actually do this through nasoenteric tube that can actually be attached with a camera, and then you can actually take the tube and intubate the person who is actually receiving this medication, and that's what exactly they did in this particular study that is shown this in these graphs. By the way, both the graphs are this exact same thing, except for that the graph A is in the linear metric graph paper, x and y axis are linear metric axis. However, in the graph B, y-axis is in the logarithmic scale. That's the only difference. Both are exactly same data, but plotted in different types of graph papers. So in this study, what they did was they actually gave ranitidine to human subjects through a nasoenteric tube, where actually they were able to follow the site where the nasoenteric tube was actually depositing ranitidine by through a camera that was attached to the nasoenteric tube. And so the the aqueous solution, the amount that was administered in these studies was about 6 milliliters containing 150 milligrams, 150 milligrams of ranitidine hydrochloride into the stomach, shown by black circles, jejunum, colored triangles, and colon, black squares, in 8 volunteers through the nasoenteric tube. The much less extensive absorption of this type of polar molecule, ranitidine, is very polar. Molecular weight is about 313 grams per mole. The polar molecule from the colon is consistent with the idea that permeability times surface area product is much lower in the colon than in the small intestine. If you compare the absorption of ranitidine between colon and the other parts, stomach and jejunum, you see that nothing much is being absorbed from colon at all in this case. And additionally, you can notice that the absorption of ranitidine is effectively ceases or stops in terminal or decline phase in about 3 hours, right around here. I'm going to use a different color to mark that. So in about 3 hours, right there, and then in this graph around there, right? In about 3 hours, when placed in the stomach or jejunum, even though the drug is incompletely bioavailable, suggesting that the small intestine is a major site of absorption for ranitidine when it is taken orally. Now, exploring further the different causes for bioavailability. So far we've been talking about different factors that affect GI absorption. Now let's spend some time understanding what are the reasons for loss of bioavailability. Not all drugs get absorbed extensively when given orally. Some of them are have extremely poor bioavailability, and in some instances, when it is extremely poor in terms of bioavailability, then we have to devise other formulation methods to eventually get the drug into the body, because you just cannot give the drug orally. Now, this diagram actually schematically shows the areas through which, or some of the primary reasons for loss of bioavailability. A drug that is given as a solid or solution, it encounters several barriers and sites of loss during its progressive and sequential movement from the GI tract to the systemic circulation. Dissolution is as a primary prerequisite before the drug molecule can move across the gut wall. It's the first step. Incomplete dissolution, slow penetration of the gastrointestinal membranes, or decomposition in the gut lumen are all causes for poor bioavailability. Now, in this case, the diagram actually shows a typical path that a drug molecule can take when it is given orally. First of all, some part of the drug can actually pass right through the lumen into the colon and eventually excreted through the feces, right? So that is something that is lost to feces, through feces. It's excreted from the body. Some part of the drug will get absorbed. Again, sorry, I'm going to use a different color. Some part of the drug actually gets absorbed and has to pass through gut wall, the cells that line the GI tract. However, one thing to remember is there are numerous enzymes that are present in the gut wall, and these enzymes are similar in terms of scope and activity to those enzymes that metabolize drugs in the liver, and hence these gut wall enzymes can actually also metabolize, and there goes a significant portion of the drug. So the fraction that is lost or that circumvents in fecal loss or fecal excretion is actually labeled F sub F, and the fraction that circumvents or that is able to pass through the gut wall metabolism process is labeled as Fg, F sub G. And then past the gut wall, as I indicated in the earlier handout, everything that gets absorbed through the cell layers into the systemic circulation from the GI tract will reach portal vein, which carries the material into the liver, right? And so the liver, the portal vein takes everything to the liver, where again there's another opportunity for drug to be lost, and that is through hepatic metabolism. And the fraction that actually is capable of escaping the metabolic processes in the liver is labeled as F sub H. And so the overall bioavailability is then a product of these three things together, these three factors. Overall bioavailability indicated by uppercase F is equal to F sub F times F sub G times F sub H. Each of those fractions represent the fraction of the dose that was administered orally that was capable of bypassing or circumventing loss through feces, through gut wall metabolism, and hepatic first pass metabolism. Now, I indicated earlier that there are uptake transporters as well as different types of transporters that are spread across the GI tract. Now, there are several examples for uptake transporters. One uptake transporter is the PEPT1 or peptide transporter 1, and another is the organic anion transporting polypeptide or OATP3. These types of transporters are spread or expressed across the GI tract, and these actually have been found to facilitate absorption. Now, there are another group of transporter. These are called as efflux transporters. These actually are transporters that have been found to efflux or eject or propel drug molecules from the intracellular environment to the extracellular environment. So these transporters are actually harmful and deleterious to the overall bioavailability of molecules. When these transporters are present, it's not good for the drug molecule, because these molecules that bind to or get attached to these transporters, efflux transporters, will be pumped out from the intracellular to the extracellular environment, and hence these types of efflux transporters will eventually lead to decrease in bioavailability, and so it is a big concern, particularly efflux transporters. There are numerous examples for drugs that are substrates to this type of efflux transport. Some examples are shown there: digoxin, fexofenadine, paclitaxel, sacubitril. All these are examples for drug molecules that can actually be effluxed or pumped out from the intracellular to the extracellular environment. Additionally, exploring influence of transporters on bioavailability. Here's an example or a comparison of oral absorption between rifampicin, how rifampicin pre-treatment can actually have an impact on the absorption of digoxin. And the plots of mean digoxin concentration. Mean digoxin concentration is what is actually shown in y-axis, and the time in hours after the dose is administered over the time period which samples were obtained and analyzed for the drug is shown on the x-axis. And so the plots of the mean plasma digoxin concentration versus time profiles after oral and IV administration. The IV administration was given over as a 30-minute infusion for 1 milligram digoxin alone that is the black circles. And after 10 days of rifampin treatment, 60 milligrams daily, that data is shown in red circles to 7 healthy adults. When digoxin was given alone, as you can see um, by the black graph, uh, there's definitely a greater amount of drug that has been absorbed. But after rifampin pretreatment, the absorption of digoxin drastically reduced. And this type of influence is due to the fact that rifampin has an impact, has an effect in terms of enzymes. And in this particular study, what they actually found was digoxin molecule, the bioavailability, which is typically about 70%, was drastically reduced. And the rifampicin treatment, rifampicin when given as a pre-treatment during the study, actually led to a decrease, and digoxin is also given orally. Okay, now when it was given as an infusion, which is the data actually that is shown in a right graph that I marked with an X, the absorption is not impacted. Rifampin did not seem to impact the absorption at all, and that's because of the fact that obviously when the drug was given, digoxin was given as an infusion, obviously it did not have to go through the GI tract where rifampin was exerting some of the effect that led to decrease in bioavailability of digoxin. And so why or how this happened is that rifampicin primarily they actually studied Cmax and the Tmax parameters, and the mechanism here that explains the drastic differences that is shown between the oral and the intravenous treatments is that there's an increase in intestinal expression of P-glycoprotein, PGP, which is a very common efflux transporter, and hence P-glycoprotein being an efflux transporter leads to pumping out of digoxin from the intracellular environment. Rifampin produces overexpression of this, and when there's overexpression of this in the intestinal tract, then it leads to efflux or removal of digoxin from intracellular to extracellular and into the lumen, and hence loss, and that contributed to the loss of bioavailability in a case of digoxin. Okay, we'll stop there today. I will continue this lecture as a second video to be followed.